Cancer is a prevalent and devastating disease, affecting millions of people worldwide each year. It is a leading cause of death globally, responsible for nearly 10 million deaths in 2020, roughly 1 in 6 deaths. The most common types of cancer include breast, lung, colon, rectum, and prostate cancers. The impact of cancer is profound, extending beyond mortality to affect the quality of life for patients and their families. It brings physical suffering, emotional distress, and financial burdens due to high treatment costs. Early detection and effective treatment can greatly improve outcomes. Cancer is characterized by the uncontrolled growth and spread of abnormal cells. Anti-cancer medicines, also known as anti-cancer or antineoplastic drugs, are vital in its treatment. These medicines target and destroy cancer cells or inhibit their growth while striving to minimize damage to healthy cells. Advancements in anti-cancer medicines have greatly improved survival rates and quality of life for many patients. Approximately 25% of modern Western pharmaceuticals are derived from ingredients found in tropical rainforests. Here are 10 widely used anti-cancer drugs that originate from rainforest plants. 1. Taxol. Taxol is widely used to treat breast, ovarian, and lung cancers, as well as Kaposi's sarcoma. It works by stabilizing microtubules, preventing cancer cells from dividing, and halting cancer cell proliferation. This drug has saved countless lives by becoming a cornerstone in cancer chemotherapy. Taxol was discovered in 1962 during a National Cancer Institute program that screened plants for anti-cancer properties. Monroe Wall and Munsuk Wani isolated paclitaxel from the bark of the Pacific yew tree in 1971. Due to environmental concerns about harvesting yew trees, a semi-synthetic method was developed in the 1990s. The Pacific yew tree, scientifically known as Taxus brevifolia, is a small evergreen conifer native to the Pacific Northwest of North America. It typically grows to heights of 10 to 15 meters and has a slow growth rate. The tree is known for its thin, scaly bark and dark green lance-shaped leaves. It thrives in moist environments and can sometimes take the form of a shrub. One of its most notable features is its bark, which contains a compound called taxol. The tree grows slowly and must be around 100 years old before its bark can be harvested. 2. Homoharingtonine. Homoharingtonine is used to treat chronic myeloid leukemia. It inhibits protein synthesis in cancer cells. It offers hope for patients resistant to traditional chronic myeloid leukemia therapies. Homoharingtonine was extracted from the Cephalotaxus fortunei tree in the 1970s by Chinese researchers. It was later developed into omacitaxine mepisuccinate for leukemia treatment. The Cephalotaxus tree, also known as the plum yew, is a genus of evergreen conifers native to the temperate regions of East Asia. These trees, or shrubs, typically grow to heights ranging from 1 to 10 meters, with some species reaching up to 20 meters. They are recognized for their soft, linear leaves and drupe-like seeds that resemble plums. The Cephalotaxus species thrive in rich, forested environments, often in well-drained soils, and are valued for their ornamental beauty and potential medicinal uses. The genus has been the focus of pharmacological research, especially in the discovery of homoharingtonine. 3. Camptothecin. Camptothecin is the precursor for anti-cancer drugs like irinotecan and topotecan, which treat colon, ovarian, and small cell lung cancers by inhibiting DNA replication and causing cancer cell death. Its derivatives have become some of the most important drugs in modern oncology. Camptothecin was discovered in 1966 by Monroe Wall and Munsuk Wani from the Camptotheca tree native to China. Its derivatives, irinotecan and topotecan, were developed later to improve solubility and reduce toxicity. The Camptotheca acuminata tree, commonly known as the happy tree, is a species native to the rainforests of China. Typically reaching heights of up to 20 meters, the Camptotheca acuminata tree is characterized by its broad leaves and small, clustered flowers. It thrives in moist, well-drained soils and is often found in rich, forested environments. This deciduous tree is renowned for its medicinal properties and has played a pivotal role in modern oncology. 4. Vincristine. Vincristine is essential in treating pediatric leukemia, Hodgkin's disease, and neuroblastoma. It disrupts microtubule formation, halting cancer cell division. This discovery transformed pediatric cancer care, significantly improving survival rates. Vincristine was isolated in 1961 by Robert Noble and Charles Beer from the rosy periwinkle plant native to Madagascar. Its discovery was accidental during research on diabetes. 5. Vinblastine. Vinblastine treats Hodgkin's lymphoma, testicular cancer, and bladder cancer. Similar to vincristine, it inhibits microtubule assembly, stopping cancer cell replication. It remains a critical component of combination chemotherapy protocols. Vinblastine was also isolated by Noble and Beer in 1958 from the rosy periwinkle. It was one of the first vinca alkaloids to be used in chemotherapy. The rosy periwinkle, scientifically known as Catharanthus roseus, is a flowering plant native to Madagascar's rainforests. Renowned for its vibrant pink or white blossoms, this perennial beauty thrives in tropical climates and moist, well-drained soils. Beyond its ornamental allure, the rosy periwinkle holds a place of high esteem in the realm of medicinal botany, having revolutionized cancer treatment with its potent alkaloid compounds. It has yielded crucial anti-cancer compounds such as vincristine and vinblastine. 6. Ellipticine. Ellipticine has shown potential in treating breast and ovarian cancers by intercalating with DNA and inhibiting its replication. This alkaloid is under active research for development into effective cancer therapies. Ellipticine was first isolated in 1959 from the Acrosia elliptica tree. Robert Burns Woodward synthesized it later that year. The Acrosia tree, also known as Acrosia elliptica, is a small evergreen tree or shrub native to northeastern Australia and parts of the southwest Pacific. The tree typically grows to a height of about 6 meters, 20 feet, and thrives in tropical and subtropical climates. It is often admired for its glossy, leathery leaves and clusters of fragrant yellow or white flowers. These flowers bloom from late summer into winter, adding a touch of elegance to its appearance. One of its most striking features is its bright red, oval-shaped fruits, which are borne in pairs and resemble elongated tomatoes. However, these fruits are highly poisonous, as is the sap of the tree. Despite this, the Acrosia tree is widely cultivated as an ornamental plant due to its vibrant fruits and dense foliage. It is also used in traditional medicine in some regions. 7. Etoposide. Etoposide is used to treat testicular cancer, lung cancer, and lymphomas. It inhibits DNA topoisomerase II, causing DNA strand breaks and apoptosis. Etoposide is a core drug in many chemotherapy regimens, improving treatment outcomes for aggressive cancers. Etoposide, derived from podophyllitoxin, was first isolated in 1880 from the American mayapple plant. Developed in the 1960s by Hartmann F. Stahelin, etoposide has become an essential component in many chemotherapy regimens. 8. Podophyllitoxin. Podophyllitoxin, a precursor for anti-cancer drugs like etoposide and tenofovir, is used topically to treat genital warts and molluscum contagiosum. It disrupts microtubules and inhibits cell division, targeting rapidly dividing cells with its cytotoxic properties. Podophyllitoxin was isolated in 1880 by Valerian Pawisotsky from the mayapple plant. It has been used in traditional medicine for centuries and is the basis for drugs used in chemotherapy for various cancers. The mayapple plant, also known as Podophyllum peltatum, is a perennial herb native to the deciduous forests of North America. It is easily recognizable by its large, umbrella-like leaves and solitary white flower, which later develops into a small, apple-shaped fruit. The plant has been traditionally used for its purgative and topical applications. The compounds derived from the mayapple plant include etoposide and podophyllitoxin, which are vital in the treatment of various types of cancer. Podophyllitoxin is also utilized in the topical treatment of genital warts and molluscum contagiosum. 9. Berberine. It has antimicrobial and anti-inflammatory effects. Berberine demonstrates anti-tumor activity in laboratory studies, especially for gastrointestinal cancers. It inhibits cancer cell proliferation and promotes apoptosis, cell death. While not yet a standard anti-cancer drug, berberine has shown promise as an adjunct therapy in cancer care. Berberine was used in traditional Chinese and Ayurvedic medicine for centuries. It was first isolated in 1917. Berberis, commonly known as barberry, belongs to a genus of deciduous and evergreen shrubs found in various regions, including temperate and subtropical areas across the world. The plant is characterized by its spiny shoots, small yellow flowers, and red or blue-black berries. Berberis has a long history in traditional medicine, particularly in Chinese and Ayurvedic practices. The plant's alkaloid-rich roots and stems are often used to treat a variety of ailments, ranging from digestive disorders to skin conditions. Its most notable compound, berberine, has been recognized for its antimicrobial and anti-inflammatory effects. Berberine is also known to inhibit the proliferation of cancer cells and promote apoptosis, providing a promising adjunct therapy in the ongoing battle against cancer. 10. Lapicol. It has been traditionally used for infections and inflammation. Lapicol also shows potential anti-cancer effects, particularly in inhibiting tumor growth and metastasis. While not yet a widely used drug, Lapicol's potential continues to be explored for cancer treatment. Lapicol was isolated in 1882 by E. Paterno from the bark of the Pau d'Arco tree. It has been studied for its anti-cancer and anti-inflammatory properties. The Pau d'Arco tree, also known as Tabebuia avellanidae, is native to the lush rainforests of South America. Renowned for its strikingly beautiful flowers, the Pau d'Arco tree has been used for centuries in traditional medicine. Its bark is particularly prized for its medicinal properties. Historically, indigenous communities have utilized Pau d'Arco for its soothing qualities, employing it in remedies for various ailments ranging from fevers to gastrointestinal issues. Modern research continues to unveil the tree's promise in treating infections and inflammation, and recent studies suggest potential anti-cancer benefits. In summary, these medicines highlight the invaluable role of rainforests in advancing cancer treatment. These ecosystems remain a vital reservoir of biodiversity, with many plants, fungi, and microorganisms yet to be studied. Continuous research and conservation are essential to unlock their full medicinal potential.